The next highlight, it is from Professor Frank Slack in another extremely interesting and important subject towards microRNA-based cancer medicine. By the way, David's mother passed away because of cancer. Well, actually, we are not so sure about this, and there will be 2 presentations in the law sessions and 1 in the plenary of what happened. It's a really a very sad story. People that are interested can follow the law session and tomorrow in the plenary. But that's why this field is so important. Please, Professor.
All right, good morning, Kalimera. Thank you for the invitation to be with you today, and thank you for the kind introduction. Yeah, so today what I hope to do is provide you with one example for the, in the interest of time, of how we've translated some of the basic science discoveries in microRNAs for new potential cancer therapeutics. And as we've already heard this morning, cancer is a very pervasive disease. There are millions of cancer patients around the world, millions of cancer deaths. Cancer doesn't discriminate based on age, sex, political affiliation, where you live in the world. It strikes all humans pretty much equally. The chances of getting cancer these days is about 1 in 2 for males and about 1 in 3 for females. So very pervasive disease and very difficult to treat. And we need as many different new options as possible. And what I'm going to tell you about today is how new discoveries in different classes of RNAs could provide new targets that allow us to combat cancer. So what I'm going to be, I had to change my talk a little bit because about 2 weeks ago, you might have heard that the Nobel Prize in Physiology and Medicine was awarded to 2 scientists, Dr. Victor Ambros and Dr. Gary Ruvkun, for the discovery of microRNAs. It turns out I was a postdoctoral fellow in Gary Ruvkun's lab, and some of my work was cited by the Nobel Committee. So what I thought I would do today is begin my talk with sort of some of the background for why this Nobel Prize was awarded and why the basic science discoveries that were made in the Ruvkun and Ambros labs have led us to the point of basically providing a new potential therapeutic option for cancer. So this is a fairly recent photograph taken at our Harvard Institute for RNA Medicine Symposium in April of this year. On the very far right is Dr. Wally Gilbert. We were honoring Dr. Gilbert at our symposium. He was the Nobel Prize winner for discovering a method to sequence DNA, the famous maximum Gilbert DNA sequencing procedure. And it turns out that Gary Ruvkun, shown in the middle, was a postdoctoral fellow with Wally Gilbert back in the 1980s. And then in the 1990s, I was a postdoctoral fellow with Dr. Ruvkun. In genetic terminology, we think about this as sort of a lineage of mentorship. So Dr. Gilbert is the P generation, Dr. Ruvkun is the first filial generation, F1, and I'm the second filial generation, F2. So I've been very fortunate to have the training that I did in the Ruvkun lab, and I'm going to explain just a little bit about some of the work that we did. It's fascinating biology. I think what it really illustrates is the importance of basic science for future discovery. When we went into this work in Gary Ruvkun's lab, we were really interested in a basic developmental biology problem, and that was how do stem cells know how to behave at particular times during development? All of you appreciate that we as human beings begin as a single fertilized embryo, a single cell fertilized embryo, and then by the time we're adults, we have about 200 trillion cells in our body. Those cells are produced by stem cells that are dividing as the animal, as we grow, basically providing differentiated cells that are going to be making the neurons, the muscles, the different cells in our system. But how do those stem cells know what to do at the right time So that was the question that we were trying to answer in Gary Ruvkin's lab. So this particular question led us to the discovery of microRNAs. So before I tell you about that basic science discovery, let me explain a little bit about what microRNAs are. So we've already heard today about RNA. It's the sort of stepson or stepchild of DNA. It's a very important component of the process of life that we just heard about from our previous talk. The microRNAs, as their name suggests, are very small RNAs. In fact, while a typical RNA in our cells is about 2,000 nucleotides long, a microRNA is only about 20 nucleotides long. So very small. We still don't know whether they're the smallest RNAs, possible in our cells, but at the moment they're the smallest RNAs that we know about. And yet their size doesn't mean that they're not very important. In fact, I'm going to show you in a minute that they seem to be very important and that there may be new avenues for us to combat disease. So our work and others in the field have shown that microRNAs are in fact transcribed in the process that Davis just told us about in the last talk from our own genes. So I'm not sure if this point is working so well. It's not really. But on the far right there, you can see that there are genes within our body that transcribe RNAs that go on to make microRNAs. These microRNAs are initially formed in a way that they can base pair with each other to form that structure that looks a little bit like a hairpin, and in fact we call it a hairpin loop. And that hairpin loop structure then finds its way into the cytoplasm of the cell, shown here in the middle, where it engages a protein complex called the RNA-induced silencing complex. When the microRNA enters the RNA-induced silencing complex, it guides that complex to other RNAs that are complementary to the microRNA, so that these microRNAs base pair with other RNAs, and those RNAs then get shut down in their translation, or maybe they get destabilized so that those target genes are no longer made at the right levels. So the work that we were doing in Gary Ruvkin's lab used a very simple model organism called C. elegans. One reason why I'm presenting this introduction is that you probably, if you've read the Nobel Prize award announcement, there are some pretty esoteric terminology in that announcement, talking about C. elegans and stem cells and development, and a bunch of genes called LINs and LETs, and I'm going to explain what some of those genes are to you today. But in the C. elegans system, this is a very small nematode. It's only about 1 millimeter in length. It has only 1,000 cells. So even though it's very simple, very small, it has most of the same cells that we do in our body. We just have many more of them. It has neurons, it has muscles, it has intestinal cells. What makes it such a fantastic advantage to work with in the lab is that it has a very short lifespan. It goes from being a single cell embryo to being an adult and dying within about 3 weeks. So we can do experiments very quickly in this organism. We can mate them, cross them. We can actually watch the cells dividing as the animal grows. And then we can watch these stem cells behave. We can watch the cell divisions at every point during the development of this organism. And it goes through 4 larval stages. At each larval stage, the animal molts its cuticle and grows a little bit bigger, and then it gets to the point where it needs to molt the cuticle again and grow again. And then at the adult stage, the animal stops growing and produces its progeny. So using genetics, and this was in the sort of early 1990s, the Ruvkin and Ambrose lab, including the work that I did in Gary's lab, had produced a pathway of genes that were required for stem cells to do the right thing at the right time. We knew, for example, that there were genes that were acting in the first larval stage to the second larval stage that were required for example, telling the stem cells to do the next step in their development. And I'm going to give you an example of what that looks like at the level of cells in the next slide. And we knew that this pathway existed for all of the larval stages. And you can see that some of these genes had names like LIN4 shown in blue, LET7 shown in blue. The lin stands for lineage defective. So in a mutant that has a mutation in the lin4 gene, the stem cells have the wrong lineage going forward. I'll show you an example of what that looks like. In the case of LET7, the actual effects of that lineage problem lead to the animals dying. So the LET stands for lethal. This is what these animals look like if you just look at them under the microscope. Shown here on the left is a normal adult animal on the top called wild type. In the lin4 mutant, these animals actually accumulate extra cells. They grow bigger, and I'll show you why that is in a minute. It's because the stem cells continue to divide when they should have stopped dividing. Another mutant called lin14 is shown on the bottom. You can see it looks a little bit smaller. Hopefully you can see that. It's smaller because the stem cells have stopped dividing too soon, and the animal has too few cells. And by genetics, you can see that lin4, we knew negatively regulated the lin14 gene. That's just done by a pure genetic epistasis experiment, which I'm happy to explain to anybody over the coffee break. In my work, shown on the right, I had identified and cloned a gene called LET7, which we knew negatively regulated a gene called lin41. And similar logic applies in both of these cases. So in Victor Ambrose's lab, they cloned the lin4 gene. In Gary Ruvkin's lab, I cloned the LET7 gene. What was really interesting about these two genes was that there was no protein coding region in that surrounded the DNA where the mutations were. And so we struggled to try and determine what the actual product of these genes was. And that ultimately led us to this understanding that these genes encode for a very small RNA called a microRNA. So that's the genetic discovery of the microRNAs. That's the phenotype of the LET7 mutant. The problem with the stem cells leads to it basically bursting through an orifice called the vulva. I'll show you what that looks like in the next slide. All the animals will die. So this is a transmission electron microscope image of a C. elegans animal. Hopefully you can see the head, basically indicated by the mouth. The orifice through which the organism bursts in the LET7 mutant is called the vulva. It's shown there in the middle. And then what you're looking at is mostly the cuticle surrounding this animal. Now, this cuticle is produced by skin cells that are populated by stem cells in the skin. And I'm going to show you what some of the stem cells look like in a minute. But basically, underneath that ridge that you can see running along the top of the animal are 16 stem cells aligned in a row all the way across the animal. And those stem cells will divide with a particular pattern at each larval stage. So that's shown over here. So just looking at one of those stem cells, the V1 to 4 cell, you can see that at the first larval stage, that skin cell divides with a pattern that produces a stem cell that goes on to divide again later on, and a terminally differentiated skin cell. At the second larval stage, the stem cell duplicates itself. It makes 2 stem cells, and that goes on to then produce a stem cell and a terminally differentiated cell. And that pattern is basically what you see for the normal stem cells. In the lin4 mutant shown in the middle there, you can see that the stem cell fails to ever do anything different than what it was doing in the first larval stage. It never knows to go into the second larval stage or the third larval stage. So it's doing the wrong thing at the wrong time. In the LET7 mutant shown just next to the image, you can see that everything looks normal until the adult stage, when that stem cell should have stopped dividing, but instead it continues to divide again. And so we knew from looking at these phenotypes that maybe, if these genes were important and if these genes were found in humans, that maybe they could have something to do with cancer. Because as you know, cancer is a disease of proliferation, excess cells. Basically, cells don't stop dividing, they continue to divide. That turned out to be the case, as I'll show you in a minute. So when we cloned the LET7 and the lin4 genes shown here on the left, we were able to show that, in fact, the RNA that comes from these genes made a stem loop structure that looks something like this. That stem loop gets processed by a series of enzymes to make just the mature sequence shown in the gray outline. And it's the mature sequence, that 22 or 21 or 20 nucleotide RNA, that's the active ingredient of a microRNA gene. That is the piece of the RNA that guides the risk complex to negatively regulate the other downstream target genes. Now, why this discovery of LET7 turned out to be so important was that we found very soon after cloning the LET7 gene in C. elegans that in fact humans had a copy of LET7. Not only that, in fact, humans have 14 copies of the LET7 gene so we have 14 copies of LET7 gene in our genome, and it's exactly 100% conserved from what you see in C. elegans. So over the course of whatever, 650 million years of evolution, the LET7 gene hasn't changed. And that tells us that it's an extremely important gene. This discovery then prompted myself and others in the field to go looking for other microRNA genes. We figured if there are 2 in C. elegans and there's some in humans, maybe there are many more of them. We now know that there are in fact 2,500 microRNA genes in our genome, and almost every multicellular organism makes microRNA genes, or has microRNAs made from their genes. The other part of the story that was noted by the Nobel Prize was not just the discovery of microRNAs, but also their mechanism of action. And this is where the genetics turned out to be pivotal again, because we knew from the genetics that LIN4 negatively regulated LIN14, and we knew that LET7 negatively regulated LIN41. So we were able to ask, well, how does that negative regulation occur? And it turns out it occurs by base pairing. So in the case of LIN4 and LIN14, shown in the middle there on the right, the LIN4 sequence is shown on the bottom, and it base pairs with the LIN14 message through one of these sites that's shown here in the 3' untranslated region of the messenger RNA. And in the case of LET7 shown on the bottom, you can see that there's also complementarity between LET7 and the LIN41 gene. So this provided the mechanism of action. It said that these small RNAs are in fact binding through very small bits of base pairing and complementarity to downstream target genes. And we now know that this is a feature of microRNAs. They're all produced in this particular fashion with a hairpin structure, and they all bind through partial complementary sequences to downstream target genes. And I told you that there are 2,500 microRNAs in the human genome. We now know that almost every human gene is regulated in a similar way by microRNAs. So these are critical regulators of gene expression or gene use in our bodies. And they're made in all of our tissues. Some of them are made more highly in neurons, some are made more highly in muscles, some are made more highly in skin cells. But what this has meant also is that we can now ask whether these are involved in disease. If they're involved in normal development, maybe they're also involved in disease. And in fact, that is true. So that's been one of the discoveries that my lab has made over the last few years, is that microRNAs, in fact, regulate many disease genes. And if they regulate disease genes, well, then maybe we can use them as a way to manipulate the levels of disease genes in a beneficial way. And I'll tell you about just one example of that today. But before I go there, let me also tell you that the fact that we now know that there are thousands of these microRNA genes in our genome has prompted the field to also ask whether there are other RNAs that are found in our genome that we didn't really appreciate the value of. And we now know that, and it's a little bit cut off, I told you that there are 2,500 microRNA genes, but now we know that there are hundreds of thousands of new RNA genes that our bodies use, and that we didn't really appreciate it until maybe 15 years ago. So maybe the last decade is really when it's really exploded. So what I hope you will take home from my talk today is that the one example that I'm going to give you is just the tip of the iceberg of where we think this work could lead to in the future. I'm going to tell you about one microRNA from those 2,500, but think about the hundreds of thousands of other RNAs that are made in our bodies and how we might be able to use those in the future as well. And hopefully you'll appreciate that we're on the cusp of I think, the next breakthrough in medicine, and that is targeting these RNAs as a way to treat disease. So of course, there's a lot of excitement now around these non-coding RNAs. They're called non-coding because they don't produce proteins. They're RNAs that aren't acting as messenger RNAs. We now know that they're a new paradigm for gene regulation. The Nobel Prize recognized this 2 weeks ago. But the part I'm going to talk about mostly is this part on the right, and that is the new paradigm for diagnostics and therapeutics. So these RNAs are produced in all of our cells. They're misproduced in diseases, and we think that we can use them as diagnostics and we can target them as new therapies. So of course, RNA medicine is undergoing a renaissance at the moment. Many of you are aware of the fact that many of us took a messenger RNA vaccine for COVID, and of course, there are now messenger RNA vaccines coming for cancer and all kinds of other diseases that we'll all be taking in the future. We know of oligonucleotides that are being used to treat neurological diseases, shown on the top right. And then, of course, this is the CEO or the ex-CEO of one of the largest companies in RNA medicine, Alnylam, who now have about 6 or so RNAi drugs, siRNA drugs that are used to treat a variety of diseases. So we're entering sort of a renaissance period for this kind of research. One last thing, just very quickly, just to point out that because RNAs usually act by base pairing, if you know what the nature of the disease is, or the nature of the mutation within a diseased patient, you can sometimes design an RNA drug that's specific just for the mutation found in that particular patient. And this is an example published in New England Journal of Medicine, where an oligonucleotide drug was designed to treat a neurological disorder in one child. As far as the researchers knew, this child was the only one in the world that had this particular mutation. Designed an oligonucleotide drug that was able to prevent further progression of the disease in this patient. And so we're getting to the point of real personalized medicine that's made possible by RNA medicine. So there are many opportunities now. I'm not going to be able to talk about all of them today. I'm really going to talk about the middle one, where the RNA itself might be druggable. So the RNA causes the disease, and maybe you can design a strategy to target that particular disease. But there are many strategies that are made available by this new science. I'm going to tell you about microRNAs in cancer, although much of the economic excitement or the biotech excitement around microRNAs at the moment is in cardiovascular disease. Many of you might have heard that Novo Nordisk, the big diabetes company, just purchased a European company called Cardio for $1 billion. Cardio's main product is a microRNA that is useful in treating cardiovascular disease, or in fact, heart disease. But in our case, I'm going to tell you about one example in cancer. On the far top right, maybe in the middle of that microRNA microarray, you can see a microRNA called miR-155, HSA miR-155. The red boxes there indicate that this particular microRNA is highly upregulated in a particular type of cancer called ABC, diffuse large B-cell lymphoma, the activated B-cell class of diffuse large B-cell lymphoma. And so we and others wondered, well, is this microRNA gene just a marker for that disease? Could it be a diagnostic for that disease? Or could it be causal in the disease? Like, was it actually making patients get diffuse large B-cell lymphoma? So one way that we test these ideas in my field is something that you've already heard about today, and that is we build mouse models. We ask, can we express a particular human gene, or maybe the mouse version of that gene, in a mouse, and does the mouse get the disease? And so in fact, that's what we did in this particular case. Sorry, before I go there, let me just tell you that I'm going to tell you about one example today, and that's the example of miR-155 in diffuse large B-cell lymphoma. But just note that in the clinicaltrials.gov database, using a keyword microRNAs, you can see that there in the middle there are 1,355 clinical trials happening around the world with this new kind of modality. So while I'm going to give you one example, there are many other examples that are hopefully going to lead to new therapies for multiple different diseases. Okay, so the way that we tested whether miR-155 was in fact causal in lymphoma was we expressed the gene at high levels in a mouse. We know that human patients have about 50 times more of miR-155 in their tumors. We made a mouse that expressed about 50 times more miR-155. And hopefully you can see here on the very left that this mouse has a very large lymph node growing out of its neck. This is an axial lymph node. Some of you have already experienced this yourself. If you have a toothache or an infection in your mouth, this lymph node grows a little bit. And you know, your doctor's often poking you around the neck to see whether you have infections in the neck. That's the axial lymph node, but it's also the one that can grow in lymphoma. This mouse, when we asked a pathologist to look at it, he told us in the middle there that it was follicular lymphoma that progressed to diffuse large B-cell lymphoma. So that told us that this microRNA, one of the smallest genes in our genome, is in fact causal in cancer. It's causal in this particular type of cancer. And more importantly for us was that when we switched off the activity of this gene, either using a genetic trick where we can turn off the gene by adding a drug because the gene is inducible with that drug, or by designing an antisense oligonucleotide shown in red in the far right there, that in fact binds to the microRNA and inhibits it, we can see that the mice are completely cured by this treatment. So this microRNA is not only causal in the disease, but the disease requires the microRNA for its continued, for the disease. And this feature, which is called oncogene addiction, is the main reason why cancer therapies are so successful in the clinic today. The cancers depend on particular genes being present and being used, and if you can provide a drug that can hit those particular genes, the cancers can no longer survive. And this is the concept of oncogene addiction, and it told us that maybe this microRNA could be a good target for cancer therapy. And so we teamed up with, if you could maybe start the next movie on the right as well. But this is a mouse that where those B cells in the diffuse large B-cell lymphoma have completely metastasized. The mouse has now become paralyzed because it's got B cells all over its body and the brain and the spinal cord. Would you mind starting the movie on the right? Sorry, AV guy. Yeah, thank you. Okay, there it goes. It's good. Yeah. So what you can see on the right hand side is that the mouse treated with this antisense oligonucleotide. This is the same mouse just a few days later. Is completely fine running around the cage. Now we have a healthy appreciation in the biology field that treating cancer in a mouse is usually more easy than treating cancer in a human. And one reason is that humans are very heterogeneous. We all have different DNA. Cancer is a disease of our DNA. And so we had to really try to prove this idea by looking in humans. And so we teamed up with a biotech company called MicroRNA Therapeutics that had a clinical trial with a drug called MRG-106, shown in the middle there. MRG-106 is also called cobamazine, and it's a drug that targets miR-155. We went into the clinic with this particular drug for different varieties of lymphoma. This is the paper that we published that shows that this antisense miR-155 actually works really well in diffuse large B-cell lymphoma. So shown on the far right is just to tell you that this particular cobamazine drug, which is an anti-miR-155 oligonucleotide, went into phase 1 and phase 2 trials for cutaneous T-cell lymphoma and a phase 1 trial for diffuse large B-cell lymphoma. So while the number of patients that were treated is somewhat low, what we found, and shown on the bottom there, is that of the patients that were the activated B-cell type, which is the type that has high levels of miR-155, 3 of those patients had the activated B-cell type. And 1 of those patients had a complete response. So we were encouraged that this particular strategy might work for lymphoma. So why is this particularly useful information or important information? Well, it turns out that patient that responded to our drug had failed every other treatment that was provided to her for her disease. The patient came on to the trial after failing 6 different therapeutic regimens. And this is a common feature for diffuse large B-cell lymphoma. The patients have no real therapy. Even CAR T cells, which you'll probably hear about more in this meeting, a very new particular and potent therapy and immunotherapy had failed in this particular patient. We found, and shown here by the blue and the black graphs, is that both of the lymphomas in this patient completely regressed after just 2 injections of the drug into her skin. Unfortunately, what you can see is that even though this patient had a complete response, there was a residual population of cells in this patient that were resistant to the drug. And basically, those lymph nodes started to grow out in the next few months. Ultimately, this patient passed away after being taken off the trial. So I'm going to end my talk just by saying that while we're in the early stages of using this new paradigm, this new idea of RNA medicine for cancer, we feel like we're getting close. We're getting closer to potentially having a treatment for patients that are failing all other treatments. But we do know that we need to do better, and we need to improve not only the efficacy of the drug, we need to improve the ability to target this drug to the right places. And we're getting to the point in collaboration with biomedical engineers, with chemical engineers, with organic chemists to actually hopefully get us to the point of having a suitable treatment that will be really helpful for patients with cancer in the future. So my conclusions, I'm just going to go through it quickly. Obviously, cancer is very common and needs better therapies. We think that personalized medicine is showing some success with these targeted therapies. MicroRNAs are tiny regulators of other genes, including cancer genes, and they're altered in cancer. We have means to manipulate these microRNAs and use them as therapeutics. There are clinical trials, and hopefully they'll provide cancer therapies in the near future. They can be both companion diagnostics and therapeutic targets. So I told you in diffuse large B-cell lymphoma, miR-155 is both a diagnostic and potentially a therapeutic. And so we call those theranostics. I want to end just by saying that this is just one class of non-coding RNAs that are transcribed in our genome. We now have hundreds of thousands of new RNAs that we don't really understand what they do yet, but could they all be potentially targetable in cancer? So I'm going to end just by saying, what is next? I mean, this is really the tip of the iceberg for our field. We need many, many more people to try and understand what all these RNAs are doing and potentially target them for new therapies in cancer. So I'm going to end just by thanking the people who did the work in my lab. I'll thank my collaborators down here on the bottom. One thing that I wanted to point out, and I forgot to mention earlier, is that I'm a developmental geneticist by training. So I've had a lot of help from biochemical engineers and organic chemists and computational biologists that have helped me along the way. And my funding sources, including a small grant from Miragen Therapeutics to help us do the mouse work and the clinical trial, and you for your attention. Thank you very much.
Thank you, excellent. Let's see if we can come a little bit here. Do you want to know a secret? He was the first author on the article for which the Nobel Prize was given this year. He didn't mention it, but I have my information. But, sorry, it's correct, yes? Yes, it's correct. So, we have almost a Nobel laureate here. Really, this is, I remember during the ceremony in the church for the autonomy and Debra, Davis' mother, I mentioned that I gave Davis a challenge to make medicine, to change medicine from trial and error to a trial and error method. I even don't call it science, to a real science. And here, from this session today and from other sessions coming, we see that the medicine is going toward the real science. And we hope one day we do not have two Academy of Science, one Academy of Science and one Academy of Medicine. It is just one science academy. It does not make sense to have Academy of Science and Academy of Medicine. It's science. And the medicine today is basically a trial and error. And this is completely different. And we see the light at the end of the tunnel. And we'll have other presentations here because medicine today is complex. It's why we started with this subject. I'm not going to say what we need, but we have many presentations, even in terms of malpractice in the law session and the drug use and drug misuse in cancers. So probably you might be interested to hear about that. We have a coffee break now, but it was an excellent session. I'm receiving so many positive from our distribution and even from people in this room telling us this has been an excellent session. So thank you very much for all authors.